As far as inducible operon is concerned, we know that the example is lac operon or lactose operon. So it is an example of inducible operon. Now, lactose induces, as I have discussed in the previous section, that the repressor is actually being it actually is getting bound to the your depending on repressor is bound is getting bound to the operator region, and the binding of the repressor to the operator region is totally dependent on the effector molecule. That means repressor is effector dependent. So in case of inducible operon, the effector molecule is lactose, and in case of your repressible operon, the effector molecule is co-repressor. So here lactose is the your effector molecule, where in case of lac operon. Now this lactose is inducing the transcription of structural genes. Okay. So if we discuss about the lac operon, there are three structural genes. One is lac Z gene, lac Y gene, lac A gene. Now lac Z gene is actually encoding beta galactosidase. And what is the function of this beta galactosidase? Actually, this beta galactosidase is metabolizing the lactose into glucose and galactose. Got it? Now, second one is your lac Y gene, which is known as which is encoding galactoside formase. Now, galactoside, what is the function of the galactoside formase? Now, galactoside formase is actually promoting the entry of your lactose into the cell. Got it? So, one is beta galactosidase. It is actually hydrolyzing the lactose into glucose and galactose, whereas the lac Y gene, which is encoding the enzyme galactoside formase, this formase is actually allowing or promoting the entry of lactose into the cell. But as far as the function of lac A gene, your thiogalactoside acetyl transfer is concerned, which is encoded by the third gene lac A gene, its function is not yet known. Okay, so we can simply remember that three structural genes are there, lac Z, lac Y and lac A. Okay, then another in the operon there is another one called regulatory gene. So here the I gene, your in case of lac operon, codes for a repressor protein. Okay, I gene codes for a repressor protein. Now what is happening in case of lac operon? So when lactose is absent in the medium, as I have told you, when lactose is absent in the medium, because in case of lac operon, the main purpose of transcription is to synthesize enzymes which will ultimately result in the breaking down of lactose. Now when lactose is absent in the medium, there is no need of synthesizing these enzymes. Synthesizing the proteins, and which will ultimately give rise to enzymes. So there is no need of that. So no need of transcription. So what will happen when lactose is totally absent in the medium? The repressor, which is encoded by your repressor protein, which is encoded by I gene, is directly binds to the operator region. Got it? Now when it binds to the operator region, it blocks the RNA polymerase from transcribing the structural genes, because you know that operator is present between the promoter and the structural genes. So it will block the RNA polymerase from transcribing the structural genes. Now thus, as the your RNA polymerase cannot move, so no mRNA will be synthesized, and thus ultimately there will be no protein synthesis. So, and ultimately there will be no enzyme synthesis. So in a single word, I can say that absence of lactose is your switching off the process of transcription of enzymes, which transcription of your proteins, which will ultimately result in the formation of enzymes, and that had there been lactose, those enzymes would have helped in breaking down the lactose. So as lactose is absent, there is no need of synthesizing those enzymes. So here the transcription has come to an end or has stopped. But when lactose is present, so when lactose is present, there is requirement for the enzymes to be synthesized, as a result of which these enzymes will be used in metabolizing the lactose. So what will happen? Some of the lactose are transported into the bacterial cell, okay, and are converted to allolactose. So some of the lactose are transported into the bacterial cell and are converted into allolactose. Now the allolactose binds to repressor. Remember, repressor is being encoded by I gene. So the allolactose binds to repressor and prevent it from binding to the operator. So when lactose was absent, totally absent, the repressor was easily going and getting itself attached to the operator region. But now when allolactose is present, it is preventing the repressor from binding to the operator. Now as the your repressor is unable to bind to the operator region, the transcription continues and the proteins and enzymes are synthesized. Okay, and these enzymes will ultimately metabolize the lactose, which will be used up by the cell. After all the lactose has been used up, okay, after all the lactose have been used up, the repressor becomes free. Okay, lactose has already been used up, so there is no need of formation of further enzymes. There is no further formation of, there should not be any formation of enzymes, because it will cause only wastage of energy. So in that case, when lactose has been totally used up, the repressor becomes free now, okay, from the inducer molecule. Now it gets attached to the operator and switches off the process of transcription. Okay, so here we can say that presence of lactose induces the synthesis of enzymes involved in its own metabolism. So lactose is determining whether there will be enzymes for its metabolization or not. Okay, when lactose is absent, there will be no enzyme formation, and when lactose is present, there will be enzyme formation. This is what the inducible operon is. We can see this in this following diagram. As you can see here, these are the structural genes, Z, Y and A. These are the structural genes. Okay, so here there is operator region, there is promoter region, and there is your regulator gene. Got it? Now regulator gene or I gene, what it does? It encodes a protein called repressor, and to the promoter region the RNA polymerase binds. Now this RNA polymerase, this green section is RNA polymerase. Now this RNA polymerase actually brings about the transcription, because it will move to operator region. Through operator region it will move to structural gene region and then transcribe all the genes together into a single mRNA. Got it? Now say for example, in this step there is no lactose, isn't it? Is there any lactose? No. So when lactose is absent, now the I gene has produced this yellow section. The I gene has produced what? Repressor. Now lactose is absent in the environment, so there is no need of transcription. So what will happen? Your repressor will move and gets itself attached to the what? Operator region. As a result of which the polymerase cannot move ahead. So RNA polymerase cannot move ahead. So transcription will ultimately stop. Okay, this is the first case. Now let us come to the second case. What is happening? So here lactose is present. Okay, in the environment there is lactose. Got it? So as there is in the environment there is lactose, so this lactose needs to be broken down, needs to be metabolized into glucose and galactose, which can be used by the cell. So we know that the I gene is producing your repressor protein. Now this repressor protein has been formed, so its ultimate goal would be to come and get attached to the operator. So for the transcription to continue, this one must be, there must be some mechanism to stop this repressor from joining to the operator. So what will happen? So this lactose will go will be converted to allolactose and will get attached to the your what repressor part. Got it? So we'll get attached to the repressor part. Now once the repressor and allolactose combination has been formed, this will not be able to attach itself to the operator region. So if the repressor fails to get itself attached to the operator region, the polymerase would quite easily move through the structural genes and will transcribe these structural genes into a single mRNA. As a result of which transcription will continue. So ultimately the proteins will be formed, and after that enzymes will be formed, and those enzymes will be used up in breaking down this lactose. So this is what the mechanism of inducible operon. Now let us come to the second one, that is repressible operon. So what is happening in case of repressible operon? An example of repressible operon is TRP operon, that is tryptophan operon. Tryptophan is an amino acid. Got it? So here repressor, same thing. There will be regulator gene. So regulator gene will be producing the repressor. But here the situation is a bit different. Here something different happens. What is that? The repressor, which is encoded by I gene, cannot bind directly to the operator by itself. But in the previous case, it was able to bind directly to the operator in case of lac operon. But when lactose was present, it was not able to bind itself. But here something different is happening. Here repressor cannot bind directly to the operator itself. Got it? So it requires others' help to get itself bound to the operator. Why? Because here the aim is something different. In case of inducible operon or lac operon, the aim was something different. But here the aim is something different. What is that? So here the repressor can only bind to the operator when it combines with an effector molecule, okay, which is otherwise known as co-repressor. And this repressor and effector molecule complex, okay, is able to bind to the operator. So when they get bound to the operator, there will be no transcription. Okay. So in a repressible operon, the transcription is turned on in absence of the effector molecule. But when effector molecule is present, the transcription is turned off. Got it? Just the opposite of your lac operon. In case of lac operon, when the effector molecule is present, the transcription is turned off. But when lactose is absent, the transcription is turned on. Absent, the transcription is turned off. Okay, just the opposite to the lac operon in case of repressible operon. So what is happening here? In case of TRP operon, the same thing, structural genes are found, but here 5 structural genes are found, denoted by TRPE, D, C, B, A. So 5 structural genes. And obviously adjacent to the structural genes, that is TRPE, there is an operator O, denoted by O, and a promoter P. That I will be showing in the next slide, the diagram. Okay. Now the 5 structural genes are required for the synthesis of amino acid tryptophan. Okay, all these 5 genes are required. So the regulator gene, which is denoted by capital R in our diagram, produces repressor protein. That is very well known to you. So which is called aporepressor. Here the repressor protein is called aporepressor. Now when tryptophan is absent, okay, the RNA polymerase binds to the promoter. Obviously, RNA polymerase binds to the promoter, and as aporepressor cannot bind directly to the operator, so very easily the RNA polymerase can move through it, through the structural genes, and transcribe all these structural genes, which will later help in the formation of tryptophan amino acids. Got it? So here the aim is something different. Here your transcription is required for the formation of the effector molecule or the amino acid. So transcription will happen when there will be no effector molecule in the environment. and here the effector molecule is tryptophan. Tryptophan, when is found, the transcription will not occur, and when tryptophan is absent, the transcription will occur. So it's a bit different from the lac operon. So let us consider the two events. When tryptophan is absent in the medium, what will happen? I have already said that here the aim of the transcription is to produce tryptophan. Ultimately its aim is to produce tryptophan. So when tryptophan is absent in the medium, what will happen? The RNA polymerase will bind to the promoter as usual, as there is no aporepressor to bind to the operator. Aporepressor cannot bind to the operator by itself. So even though aporepressor has been formed, it will not be able to bind to the operator region. So as a result of which, RNA polymerase can easily transcribe the structural genes, which will ultimately result in the formation of what? Tryptophan. Got it? So when there is no tryptophan in the environment, tryptophan synthesis will occur. But when there is presence of tryptophan in the environment, what will happen? So obviously there is no need for the synthesis of more tryptophan, because tryptophan is found in the environment. So what will happen? The transcription needs to be stopped. So who will stop it? Obviously the job of the repressor is to stop the transcription. Now here the repressor is called aporepressor. But we know that aporepressor requires the help of somebody else. So whose help does it require? It requires the help of the effector molecule, which we call as co-repressor here. Here the effector molecule is tryptophan. We call it as co-repressor. So aporepressor and co-repressor complex, they bind with operator and blocks the function of RNA polymerase, and hence transcription stops. Okay, so transcription is not at all required when tryptophan is found in the medium, because here the aim of the transcription is to produce tryptophan. Got it? So let us come to the diagram. So it's shown in the diagram. Here also there are presence of structural genes, operator region, promoter region, then the regulatory gene. So what happens here? Let us see. So here these part includes your structural genes, the EDCBA, there is operator region, then there is promoter region to which the RNA polymerase binds, then there is regulatory region. Okay, in both this diagram, the first one and the second one, the same thing is there. So let us see. So in the first case, when there is presence of tryptophan in the environment. So in the environment, there is presence of tryptophan. As you can see, it has been denoted by this green one. Okay, so tryptophan is present in the environment. If there is presence of tryptophan in the environment, there is no need for the formation of more tryptophan or synthesis of more tryptophan. So we know that here the transcription will ultimately lead to the formation of tryptophan. So transcription needs to get stopped. We have sufficient tryptophan, so it needs to get stopped transcription. So what will happen? The aporepressor, which is encoded by R gene, it combines with tryptophan. Got it? Now the aporepressor and the tryptophan combine to form co-repressor. Now this co-repressor complex, they get attached to the operator region. Okay, as a result of which the RNA polymerase won't be able to move ahead, and thus transcription stops here. Got it? So here the transcription is stopped because the co-repressor, that is the aporepressor and tryptophan complex, they are combining with each other, and this complex is getting bound to the operator region. Okay. Now second case, when tryptophan is found, what will happen when tryptophan is absent? So when tryptophan is absent, there is need for the synthesis of enzymes which will result in the synthesis of your tryptophan. So what will happen? Transcription needs to continue. So the aporepressor, even though it is encoded by R gene, it will not be able to get itself attached to the operator region. So obviously the transcription will continue as the RNA polymerase will move ahead without any hindrances on its path. Got it? So this is how the gene expression is regulated in case of prokaryotes. Okay, so prokaryotes are generally single celled or unicellular organisms, so they require to conserve whatever energy they have. Got it? They do not afford to waste energy. That's the reason why the gene regulation is very much essential in their case. Okay. So in addition to that, there are some other kinds of genes. Okay, these are regulated genes because they are regulated in a specific way. But there are also some other kinds of genes which we call as housekeeping genes. You just remember that housekeeping genes are actually responsible for maintaining the cell structure and function. That means they continuously work. Okay, they do not get any regulation mechanism. But these express constitutively, that is, their products are constantly produced at a fixed rate. Okay, there is no regulation, they are constantly produced. They are turned on all the time. There is no switching off or switching off condition. There is no switching off time or switching off time. They remain always in a switched on condition. Okay, they generally code, these housekeeping genes generally code for enzymes that the cell requires in large amounts and for its major life processes. These include enzymes for glycolysis, Krebs cycle, ribosomal proteins, etc. So we can say that the enzyme regulation are done to conserve energy in case of prokaryotes. So enzyme regulation is also found in eukaryotes that we will study in some other classes. But here we can remember that enzyme regulation is very much essential. Similarly, there are also some enzymes which are always in a switched on condition. Regulation means they are sometimes turned on and sometimes turned off. But in case of genes called housekeeping genes, they are always in a turned on condition. They continuously produce their products at a constant rate, at a fixed rate. Got it? So this is what your gene regulation is all about. I hope that gene regulation, you will be able to understand gene regulation in a better way. Okay, thank you.